Hello everybody, today we're going to talk about gene mutations. In order to understand gene mutations, you have to first understand how proteins are made. If you're not already familiar with all the vocabulary words labeled on this picture, I would recommend you first watch the protein synthesis video. A gene mutation is a change in the DNA sequence that is going to most often result in a difference in the protein that is produced. What causes gene mutations? Well, it can be just simple mistakes that your body makes when it is either replicating the DNA or when it is transcribing the DNA into mRNA. Although those mistakes are more common if you have been exposed to a lot of sunlight, to radiation, or to certain chemicals. Before we can talk about how specific different types of mutations in the DNA could result in changes for the organism, we need to look at this original DNA and what it would originally code for in terms of its amino acid sequence. So we're just going to look at the left-handed side of the DNA, often called the template strand. That template strand would be used to transcribe and make an mRNA molecule. The mRNA would then travel to the mitochondria, where it is interpreted 3 nitrogen bases at a time. This original mRNA reads AUG, which means this tRNA molecule with an anticodon AUC would match and drop off the amino acid methionine. Okay, so how can we use that information? Just if you don't have these manipulatives at home, is your teacher may have given you a chart something like this, where you have the DNA sequence, you write out what the mRNA would be, and then you use a device like this called a codon wheel to figure out which amino acid would be attached for each segment of mRNA. So in this unmutated DNA, the original DNA sequence, this is the amino acid sequence. This would then fold in a very particular way to come up with the overall shape of the protein. If we were to mutate our DNA, and let's say that first thymine had a mutation, and instead of a thymine we had an adenine in that 4th spot within our gene. Well, that could change things significantly. So we would no longer make this kind of mRNA because you could see that this would not occur here. So we would end up making a different mRNA sequence. The first codon is the same. So we would still end up with methionine as the first amino acid that is produced in our polypeptide chain. However, when the mRNA is read for the second codon, we are going to end up with a different tRNA molecule matching, and because it's a different tRNA, it's going to be carrying a different amino acid. In this case, it's phenylalanine. So a substitution mutation generally is going to result in one amino acid being changed. So you can see in this case here was the original DNA sequence, TAA, making this mRNA and coding for this amino acid. Once we had a substitution in the first spot, we ended up with a different mRNA sequence and a different amino acid. So this polypeptide is going to fold differently than the original one, causing a difference in the shape of the protein. There are several disorders that you have heard of before that have this type of mutation. Tay-Sachs disease is one, cystic fibrosis is another, and sickle cell anemia. All are caused by just one nitrogen base being wrong, causing the mRNA to be wrong, causing the amino acid to be wrong, causing the protein to be shaped wrong. So a substitution mutation is the result of one nitrogen base difference in the DNA sequence. It's going to change maximum one amino acid, and it can cause a change in the shape and function of a protein. However, there are a couple of places where the DNA could mutate and it would not affect the protein at all. If you look carefully at this codon wheel, you will see that some amino acids are listed more than once, arginine and arginine. So you can see that there's multiple ways the mRNA could code to get that same amino acid. So if you happen to have a mutation that resulted in the same code, you're going to end up with the same amino acid, and so therefore it's not going to change the shape and function of the protein. Let's now talk about deletion mutations. A deletion mutation is literally if we were to take one segment of the DNA and cut out a nitrogen base. So if this is my new DNA sequence, this original mRNA would not happen. Instead, we're going to have to cut out a section of the mRNA for it to replicate what would really be produced. Okay, so the mRNA is now shorter by one. And what we have caused now is it's not only a deletion mutation, but it's also going to result in a frame shift. And what I mean by that is that when the ribosome reads this mRNA, the first mRNA is read, it's going to code for the same amino acid as it did before. Okay? Second one, no problem. Third one, remember there used to be a different nitrogen base there. And instead, this one's going to be slid into place. So this is going to result in a different amino acid being added. GUG is going to be valine. But it's not only going to cause a problem there. In fact, it did happen to cause no change. GUA coded for valine and GUG also codes for valine. However, if we look at future mRNA sequence, everything after this point would be affected. Now, this particular mRNA that I made is very short. It's not true to reality. In reality, this would be a much, much longer sequence. But look at this last one. It can't even find a tRNA that is going to match because it is missing a nitrogen base. So basically, not only do we have the potential to affect that one codon, but every codon after that. And that's what we mean by frame shift, is we're shifting this frame of the ribosome and what codons it's reading ever after that point. So this is going to result in a shorter polypeptide sequence, which will affect the overall amino acid sequence and therefore affect the shape and function of that protein. All right, so let's talk about an addition mutation, which is also going to result in a frameshift. We added in one extra thymine, and so therefore we're going to add in an adenine onto our mRNA. This also is going to result in a frameshift. So as the ribosome reads each codon, not only is that first one different than the original sequence, but every amino acid afterwards is going to likely be different because every codon is different than it was before. So what is the result of the addition mutation that we caused? Well, we added one nitrogen base here at the beginning, which caused everything to shift down on the mRNA. And so every codon is now different, and that resulted in all the amino acids changing. That's going to, that's why we call it a frameshift mutation. It's because it's shifting literally the frame that the ribosome is reading the mRNA. And this is going to end up causing a drastic change in the protein shape. Because if you've got all different amino acids, there's no way it's going to fold the same way it did before.